Thank you.
So our next presenter is Dr. Ria Goswami. And Ria received a master in biotechnology from India, and she then came in the US to pursue a PhD in molecular biology, microbiology, and biochemistry at Southern Illinois University School of Medicine. After receiving a PhD, she then came at the DHVI in Dr. Sally Perma's lab for a postdoc. And her work focuses on identification of novel strategy to reduce HIV reservoir in orally infected infants, and she uses both human and non-human primate models. Today, the title of her talk is Adjunctive Therapy with an Hsp90 inhibitor potently suppresses HIV replication by targeting lymphoid cell activation and proliferation.
Thank you, Jenny, for the nice introduction. Hello, everyone. Today, I will be talking about an adjunctive therapy that suppresses viral replication by mainly targeting lymphoid cell activation and proliferation. So, despite the widespread availability of antiretrovirals, approximately 180,000 infants were newly infected with HIV in 2017. And of those, 50% infections occurred during breastfeeding. These new pediatric infections could not be prevented by the current maternal antiretroviral regimens due to the limitations such as treatment non-adherence in the postpartum period, development of ARV-resistant viral strains, and most importantly, new maternal infections that occur during the breastfeeding period. These infants who are infected during breastfeeding are often not diagnosed immediately. And when they're diagnosed, they present with very high plasma viral loads and established viral reservoirs. So at this point, even when antiretroviral therapy is initiated immediately, it requires a very long time to control their viral loads, thereby further increasing the size of the viral reservoir. As a result, these infants are forced to rely on ARV for the rest of their life and are predisposed to long-term metabolic consequences and development of ARV-resistant viral strains. Now, since rapid viral suppression and reduced size of the viral reservoir has often been associated with long-term viral suppression, we thought that including an adjunctive therapy in the current ARV regimen is necessary in these infants that will rapidly control the virus and will reduce the size of the viral reservoir by preventing further establishment. It has already been established quite well that HIV preferentially likes to infect activated target cells and therefore results in systemic immune activation, and that's how the virus spreads from one cell to another cell. So the hypothesis that we had is if we block this HIV-mediated immune activation and proliferation along with the ARV regimen, then this will prevent viral replication, and this will further prevent the establishment of viral reservoir. Our collaborators have previously shown that a cellular chaperone protein, Hsp90, is upregulated upon viral infection and are involved in major stages of viral replication. Hsp90 has also been shown to be involved in immune activation. So we thought that including an inhibitor of Hsp90 in the ARV regimen will be basically a two-pronged attack on the virus directly by blocking its replication and indirectly by blocking activation-induced cell-to-cell viral transmission. Currently, 17 different Hsp90 inhibitors are being tested in clinical trials for cancer therapy in the United States. Of those, an orally bioavailable small molecular inhibitor has been tested in humans and have been found to be well tolerated with no adverse side effects. So in this study, we wanted to, first of all, investigate the combined effect of this inhibitor and antiretroviral on HIV replication using a pediatric setting, and second, evaluate the pharmacokinetics and safety of this drug using a rhesus macaque model. So before we look at the efficacy of this combination therapy, we first wanted to look at the effect of just this inhibitor on viral replication. And for that, we established an in vitro model of HIV replication in a physiologically relevant human pediatric tonsil tissue. Tonsillar mononuclear cells were infected with GFP reporter virus and were treated with 100 nanomolar Hsp90 inhibitor starting from 24 hours post-infection, basically to mimic the delayed diagnosis during breast milk transmission. On the 5th day post-infection, the proportion of CD3 cells infected by the virus was measured by flow cytometry. Our data demonstrated that in presence of this inhibitor, there was indeed a significant drop in the proportion of CD3 cells getting infected, both by a CXCR4 tropic as well as a CCR5 tropic virus. So we were then interested in looking at the effect of this compound on cell-to-cell viral transmission. And for that, this infected and treated tonsillar mononuclear cells were co-cultured with luciferase expressing TZMBL reporter cell lines and 50% cellular infectious dose, which is the number of tonsillar mononuclear cells required to initiate infection of 50% TZMBL cells, were measured using the Reed-Munch equation. So I'm showing here the infectious viral transfer in presence of the compound, and you see there has been a reduction in when the cells were treated with this Hsp90 inhibitor, which also resulted in increase in the number of infectious tonsillar mononuclear cells required to initiate infection of the TZMBLs. So now when we know that this compound is basically reducing the proportion of the CD3 cells infected by preventing cell-to-cell viral transfer, we wanted to combine this with one of the well-known pediatric ARV regimens and then look at the effect of this combination therapy in vitro in our tonsil model. We found that when this inhibitor was combined with antiretrovirals, it resulted in a significant drop in the proportion of infected cells compared to ARV by itself. So obviously, we were interested in looking at the mechanism of this compound, the anti-HIV function of this compound. And for that, we performed RNA sequencing from the total RNA extracted from this infected and treated mononuclear cells from 8 independent tonsil donors. Our RNA sequencing data demonstrated that there was a differential gene expression in the drug-treated and the control group. So the red color here represents the genes that were upregulated, and the blue color are the ones that were downregulated. We further made our selection criteria a little bit more stringent, and we only chose the genes that showed a 4-fold difference between the drug-treated and the control group. And we performed a gene enrichment analysis, basically to identify the cellular pathways that were upregulated or downregulated in presence of this compound. We then selected the genes that were significantly altered by this compound in these pathways, and we compared them with the published HIV host factors. We found that this compound downregulates 14 genes and upregulates 1 gene that are published to interact with HIV replication. Interestingly, 9 of these genes were immunomodulatory genes and 6 of them were cell cycle regulatory genes, which confirms our hypothesis that this compound is basically preventing cell-to-cell viral transmission by downregulating immune activation and proliferation. We also validated our RNA-seq data at the protein level by showing that this compound indeed significantly downregulates CD4 activation levels. Interestingly, we also found that Hsp90 inhibitor downregulated the expression of CD4 markers on the surface of the CD3 cells by increasing the proportion of CD4 negative, CD8 negative T cells. So basically, this compound is a multifunctional anti-HIV molecule which blocks cell-to-cell viral transmission by inhibiting immune activation and proliferation, restricts new HIV infections by altering the phenotype of target cells, and also downregulates host genes that facilitate viral replication. So finally, we wanted to test the pharmacokinetics and safety of this compound in vivo using a rhesus macaque model. For that, we used the orally bioavailable Hsp90 inhibitor and administered it to 2 adult rhesus macaques and 2 infant rhesus macaques, and the animals were bled at the indicated time points. We then measured the concentration of the active form of the drug in rhesus macaque plasma, and we found that even when the concentration of the drug in infants were lower than that of the adults, but the concentration was still higher than the in vitro inhibitory concentration that we have identified to be 100 nanomolar. So basically, to summarize the data that I have shown you today, incorporating an Hsp90 inhibitor in the current ARV regimen results in viral suppression by reducing the number of available target cells due to attenuation of lymphoid cell activation and proliferation. And we also have shown that this inhibitor is quite well tolerated to be used in rhesus macaques with no adverse side effects. In future, the effect of this combination therapy on rapid viral suppression, viral reservoir size, and time to viral rebound needs to be evaluated using a relevant Hsp-infected infant rhesus macaques. With this, I would first like to thank the CIFAR retreat organizers for giving me this opportunity to present my research in front of you, my mentor, Dr. Sally Parmar, other members of the Parmar FODA lab, our collaborators, and most importantly, our funding sources. Thank you.
Questions for Ria? Okay.
Ria, that was a great presentation. Do you have any idea of what impact the inhibitor has on immune responsiveness?
So there has been a couple trials in humans, basically for cancer therapy with this inhibitor, and they did not see these patients to be immunosuppressed or having any adverse side effects, but we haven't tried that in infant macaques. That's the plan to do next year. But obviously from the in vitro data, it looks like this inhibitor will be an immunosuppressant to be particular.
But it also decreases the number of, or the frequencies of CD4, CD25 cells,
Right.
which define Tregs in certain instances. So it could also have an opposite effect. I was just interested.
Right, that's true, yeah. Hey, question?
Any other questions?
Thank you.